Welcome to the PubMed Daily Review for May 6th, 2026. Today we scanned 6,678 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Neuroscience and Pain Mechanisms. This research explores the neural basis of pain, its modulation, and its connections to neurodevelopmental and neurodegenerative conditions. This cluster covers 724 papers. Representative work includes conditioned pain modulation intersite variability study, effect sizes and test-retest reliability of 2 models, the multifaceted roles of eukaryotic elongation factor 1 alpha 2 in neurodevelopmental and neurodegenerative disorders, and amygdala and nucleus accumbens activation are associated with treatment choice in knee osteoarthritis. An fMRI study. Trend 2 of 5, clinical studies and interventions. This trend unifies research investigating clinical conditions, interventions, and patient outcomes through diverse methodologies. This cluster covers 1,539 papers. Representative work includes investigation of some clinicopathologic alterations in cats infected with Mycoplasma haemophilus and Candidatus mycoplasma haemominidum in Mashhad, Iran, an observational cross-sectional study. Efficacy and safety of the use of interspinous spacers in the treatment of lumbar spinal stenosis, mapping gap and scoping review, and a systematic critical appraisal of clinical practice guidelines for non-pharmacological conservative management of complex regional pain syndrome, CRPS, using the Agree-To instrument. Trend 3 of 5, health education and social aspects. These papers analyze social, educational, and policy factors influencing health behaviors and outcomes across diverse populations. This cluster covers 1,339 papers. Representative work includes a proposed participatory framework for explainable AI in mHealth. Mixed methods study integrating user and stakeholder requirements. Lexical analysis of terms related to nursing care for pregnant women with syphilis and pain retrained. Outcomes analysis of an online, interdisciplinary chronic pain education program. Trend 4 of 5, metabolic and infectious disease. This trend investigates the mechanisms, diagnostics, and physiological impacts of metabolic processes and infectious diseases. This cluster covers 2,127 papers. Representative work includes assessing the impact of female genital mutilation/cutting on genital inflammation and microbiota among Kenyan female sex workers. Temporal metabolomics profiling reveals liver metabolic control in single and repeated exhaustive exercise in murine models, and effects of intermittent fasting mimicking diet on pancreatic islet plasticity. Immunohistochemical, ultrastructural, and metabolic profiles. Trend 5 of 5, oncology, mechanisms, diagnostics, therapy. This trend integrates research on cancer mechanisms, advanced diagnostics, and innovative therapies for better patient outcomes. This cluster covers 949 papers. Representative work includes awake percutaneous cervical chordotomy in patients with cancer, a technical report, Pap testing, and high-risk HPV testing for women aged 65 years and older with surgical pathology follow-up, and analysis of PLEKHS1 promoter mutation in preoperative thyroid nodule samples. Up next, our first paper out of 5. Illuminating Cell States by a Comprehensive and Interpretable Single-Cell Foundation Model by Zhu Wang and Stan Z. Li from University, Hangzhou, China. Let's break down its key contributions. Single-cell sequencing has revolutionized our understanding of biology, but the sheer volume and complexity of this data pose significant challenges for AI models. Current approaches often fall short due to data sparsity, heterogeneity, and a lack of interpretability, hindering their real-world application. This paper introduces a novel solution to these critical limitations. The researchers developed CellVQ, a comprehensive single-cell foundation model. It was trained on an enormous dataset of 68 million cells, featuring a unique single-cell discretization module. This module transforms complex data into interpretable cell codes and addresses data heterogeneity, a major hurdle in single-cell analysis. They also introduced CellVQ Graph, a tool for integrating multimodal data into a knowledge graph. The results were truly impressive. In cell clustering, CellVQ achieved an NMI of 0.9424 and an ARI of 0.9492 on the pancreas dataset, far surpassing other methods. For cell annotation, It reached an accuracy of 0.88. Across all evaluated datasets, CellVQ consistently outperformed strong baselines, often by substantial margins, highlighting its robust performance and zero-shot capabilities. This study marks a significant step forward, offering a truly applicable and generalizable AI tool for the cell biology community. CellVQ's ability to provide interpretable cell codes and integrate multimodal data will accelerate biological discovery. While its performance is outstanding, ongoing validation and application to new biological contexts will be key to unlocking its full potential in understanding cellular mechanisms. Up next, our second paper out of 5, Extrachromosomal DNA Gives Cancer a New Evolutionary Pathway by Elizabeth Brunk and Logan Slade. Let's break down its key contributions. Traditionally, we've thought cancer evolves through individual cells acquiring advantageous mutations. But what if there's another way? This paper dives into extrachromosomal DNA, or ecDNA, which seems to give cancer a unique evolutionary edge. Understanding this mechanism is crucial because ecDNA-driven cancers are notoriously aggressive and hard to treat, making this research highly relevant for developing new therapies. To unravel ecDNA's secrets, the researchers employed a sophisticated array of techniques. They analyzed thousands of single cells using advanced imaging, single-cell multiomics, and multiplex proteomics. This comprehensive approach allowed them to precisely characterize ecDNA levels and their functional impact, even using stochastic models to understand the dynamics of ecDNA distribution over time. The study revealed that higher ecDNA dosage directly leads to proportional increases in gene expression, protein levels, and cell proliferation. Interestingly, genes on ecDNA showed unique transcriptional scaling compared to chromosomal genes. When the researchers experimentally disrupted the ecDNA distribution in cells, the population rapidly recovered its original continuous spectrum. This recovery wasn't random. It was an active, deterministic process, highlighting ecDNA's dynamic and adaptive nature. These findings fundamentally shift our understanding of cancer evolution, showing that ecDNA offers a distinct pathway for tumors to rapidly adapt and resist treatment. The ability of ecDNA to dynamically create a spectrum of oncogene copy numbers acts like a bet hedging strategy, ensuring tumor survival under diverse pressures. Unraveling this active, deterministic process of ecDNA restoration opens exciting new avenues for developing targeted therapies against some of the most aggressive and challenging cancers. Up next, our third paper out of 5, Multi-Hospital Electronic Health Record Foundation Models Without Data Sharing. A comparison of federated learning and Inference-Time ensembling by Olivier Elemento. Let's break down its key contributions. Foundation models for electronic health records show great promise, but current models are limited to single institutions due to strict privacy regulations. This creates a significant barrier to developing more robust, generalizable AI tools for healthcare. Our study explores innovative ways to build these powerful multi-institutional models without ever sharing sensitive patient data. To tackle this, we developed a GPT-style EHR foundation model using data from over 100,000 patients from the MIMIC-4 dataset, simulating 5 distinct hospital sites. We then rigorously compared 3 approaches: traditional centralized training, federated learning, where only model weights are exchanged, and Inference-Time ensembling, which only shares predictions. We evaluated these models using standard metrics like OROC and AUPRC for various clinical predictions. Our findings were quite compelling. While centralized training achieved the highest performance with an AUPRC of 0.376, federated learning recovered an impressive 85% of that performance, reaching 0.321. Inference-Time ensembling, which is even more privacy-preserving, still recovered 77% of the AUPRC. Crucially, an estimated 87% of participating hospitals actually received a better model by joining the ensemble than by training alone. This research clearly demonstrates that multi-institutional EHR foundation models are indeed feasible without direct patient data pooling. Inference-Time ensembling stands out as a highly practical solution, offering significant benefits to most hospitals with minimal governance overhead. While federated learning provides slightly better performance, it requires more complex weight sharing. These findings pave a clear path forward for collaborative AI development in clinical settings, promising more powerful and widely applicable tools for patient care. Up next, our fourth paper out of 5. Two-Photon 3D Printing of Functional Microstructures Inside Living Cells by Marissa Moore and Majof Humer from Department of Condensed Matter Physics, J. Stefan Institute, Ljubljana, Slovenia. Let's break down its key contributions. Imagine the precision of 3D printing, but not on a factory floor, but inside a living cell. While 3D printing has transformed manufacturing and biomedicine, the ability to create structures directly within cells has been a significant hurdle. This groundbreaking research tackles that very challenge, aiming to open up entirely new frontiers in cellular engineering and understanding. The researchers developed an ingenious method using two-photon polymerization. They injected a special biocompatible photoresist directly into living cells, then precisely used a femtosecond laser to selectively polymerize it. This allowed them to print custom-shaped microstructures with incredible submicron resolution right where they wanted them, inside the cell. The results were truly remarkable. They successfully printed structures of various shapes, including a tiny 10-micrometer elephant, barcodes for tracking, and even microlasers, all directly inside living cells. This was achieved with incredible submicron resolution, showcasing the precision of their technique. Importantly, they also observed that these printed structures could actually affect the cell's biology. This research represents a monumental leap, demonstrating the first successful 3D printing inside living cells. This breakthrough opens up a world of possibilities, from advanced intracellular sensing and biomechanical manipulation to highly targeted drug delivery. Imagine being able to engineer cellular properties in ways previously thought impossible, going beyond even genetic engineering. Of course, further research will be crucial to understand the long-term effects and translate these incredible findings into clinical applications. Up next, our fifth paper out of 5. Feature representation for explainable CRISPR off-target prediction and base editing efficiency by Faiza Hassan and Tommaso Selmi from Department of Computer Science, University of Bari Aldo Moro, Bari, Italy. Let's break down its key contributions. CRISPR, Cas9 gene editing holds immense promise, but unwanted off-target edits are a major hurdle. To overcome this, we rely on advanced machine learning models to predict these interactions. However, a critical question remains. How does the way we represent genetic sequences influence both the accuracy of these predictions and our ability to understand the underlying biology? This paper dives deep into that very question. Our study employed XGBoost machine learning models, a robust algorithm, across two key CRISPR applications, gene knockout and base editing. We utilized established benchmark datasets like ChangeSeq, GuideSeq, and the HANA screening dataset. The core of our methodology involves systematically evaluating how different ways of encoding DNA sequences, from simple one-hot to complex Bolges representations, affected the model's predictive power and our ability to interpret its decisions using tools like SHAP. Our results showed that for gene knockout, one-hot and its variants achieved the best performance on GuideSeq, with an AUPR of 0.661 and a Pearson correlation of 0.756. For base editing, one-hot encoding consistently outperformed other methods, yielding an impressive AUPR of 0.723 and a Pearson correlation of 0.746. Crucially, our interpretability analysis revealed that despite the different mechanisms, the protospacer adjacent motif, or panproximal region, remained a critical feature for prediction in both editing types. This study underscores that the choice of feature representation is not just a technical detail. It's fundamental to building accurate and, more importantly, interpretable CRISPR prediction models. While we didn't find a universal best in coding, our work highlights the need for tailored approaches based on the specific CRISPR task. Moving forward, continued innovation in feature engineering and a strong focus on model interpretability will be key to developing safer and more effective CRISPR technologies for clinical and research applications. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.